Okay, so before we get started, does anybody have any questions? Okay, well, I want to go through some of the things that are in the two chapters that we're doing this week, Chapter 6 and Chapter 7. One thing that I did want to address, somebody had made a comment on one of my other classes on the discussion board where the questions are. In my other class, somebody had made an anonymous comment about how fast they thought the class was going and how much content they thought there was for the class. And for a summer class, particularly for an online summer class, it can feel that way. Normally in the summer, you'd be spending 6 hours a week in class because we've taken a 16-week semester where you'd spend about 2.5 or 3 hours a week in class. And we kind of compress it into a 7.5 or an 8-week summer session, so that would double up the time each week. And we normally tell students to plan on spending about 2 hours outside of class for every hour that you're inside of class, trying to do, the internet research and the reading and the studying and the learning part that's the more independent piece. So it is going to seem like it's pretty fast-paced, and it is going to seem like a lot of material, especially the biochemistry stuff that we just went through is not always really easy for a lot of people. It's not something that people have been exposed to a lot, and so to do that kind of quickly it does make it really difficult. So I just wanted to let you know that I was kind of having that conversation with my other class, and that I know that it feels really fast-paced, and then to not have that in-class time, to be doing a lot more of it independently, which is going to be the nature of an online class, even kind of compounds that. But any questions that you guys have or any help that you need from me, please let me know. I'm trying to give as much feedback as I can on grading and things to kind of help shape some of the things that you guys work on in the future. So I'm hoping, that's my hope, I'm hoping that I'm providing some feedback to you guys. And if you need additional feedback, please let me know. Is there anyone that tutors? There is on the Jefferson campus, that's where I'm located, at our downtown campus at Jefferson Community and Technical College. There's a teaching and learning lab for math and natural sciences. It's in the Hartford building on the downtown campus. That's the big, tall building on the downtown campus. It's on the 5th floor, right as you get off the elevator. And they do have people who can tutor in Bio 112 in the intro biology classes. So you might, I don't think I have their number, but you might see if you can stop by there, or you might give them a call and find out. What I can give you is I have it, Natural Sciences and Mathematics Learning Lab on the downtown campus. The phone number is, it's 502 area code 213-5086. Let me type that in here so it's up here for people who need it. 213-5086. So they can put you in touch with people that are doing tutoring, people that are coming in there to have kind of office hours kinds of things. So that's a good resource for you. That's the Natural Sciences and Mathematics Learning Lab. A lot of people just refer to it as the Learning Lab, but it's the Natural Sciences and Mathematics Learning Lab. Those are some folks that can get you in touch. If you're on a different campus or at a different college than Jefferson, you can contact your local college and see if any of your local student services people or tutors are in over the summer. And if there's specific questions that you have, if there's specific questions that you didn't do well on the test or specific things about the assignments that are confusing, please feel free to send me an email, and hopefully I can I don't know that that's much more help than what's already online, because again, that's just an online interaction, and some people do well with the face-to-face interaction, with being with somebody that can explain it. But let me know what other things that I can do, and I will try to accommodate those. So that's a good question about resources. So this week, we're talking a lot about DNA. And so I put this slide as one of the first slides in the PowerPoint to kind of give you an overview, because we're going to be talking about several different things related to DNA, and then we're going to be talking about other molecules. And so to try to help you fit in how that all works together. So DNA does two things. One of the things that it does is it makes copies of itself. When the cell needs to divide, it needs to make a copy of itself. It needs to make a copy of everything that's in the cell, including the DNA. The other thing that DNA does is it has the instructions for how to make macromolecules like proteins. So Chapter 6 talks about the copying, and that's replication. If I can type that in here somewhere. Replication is the term that we use when we talk about DNA copying itself. And then Chapter 7 talks about how DNA is used to make proteins. And that process is transcription. Where DNA is used as a template in the first steps of protein synthesis. Now, we've talked about DNA briefly already. DNA is one of our macromolecules. It's a nucleic acid molecule. So it's formed by subunits called nucleotides coming together by covalent bonds. And then the two strands of the DNA are held together by hydrogen bonds. And you're going to see that again in just a second. So there's this kind of assertion or theory in biology that is often called the central paradigm of biology, or especially molecular biology. So if you search on that central paradigm of molecular biology on the internet, you'll come up with this. And so basically, the idea is that what we're talking about, the DNA can do a couple of things. And one of those things that DNA can do is DNA can make a copy of itself. DNA can use itself as a template to make a copy of itself. That's the process that's called replication. Would it help if I had typed that spelled correctly? Replication. There we go. Trying to go too fast. And then the other thing that can happen is in the protein synthesis piece, where DNA is used as a template to make a copy molecule called RNA. And then that RNA is used as a template with the directions to make a protein. The process of DNA making an RNA is a process called transcription. And then the process of RNA making a protein is a process called translation. And so those are the processes that we're going to be talking about in these two chapters. Chapter 6 talks about the replication piece, and then Chapter 7 talks about the transcription and translation piece. So hopefully that gives you a little bit of a kind of roadmap of what we're talking about. So in Chapter 6, we stick to this. DNA makes a copy of itself. and we'll be talking about how DNA makes a copy of itself. And then in Chapter 7, we'll talk about how DNA helps to make a protein. Now, we talked about DNA structure just briefly when we were in Chapter 2, so you can go back to Chapter 2 and look at some things about nucleic acids and how nucleic acids make a DNA molecule. And then this chapter goes into a little bit more detail about that structure. So basically what happens to make a DNA molecule, there are two long chains of nucleotides. Remember, all of our macromolecules are these long chains made up of these subunits. So a DNA molecule is two long chains of these nucleotides. And then they're coiled into a double helix. And that double helix is held together by hydrogen bonds. So this is going to rely a lot on some of the things from some of the previous chapters, especially Chapter 2 and Chapter 3, as we go through some of this information. Your book shows you the structure of a DNA nucleotide. It has a sugar on it called deoxyribose, that's the D in DNA. And it has a base. And there are four versions that base can be in DNA. It can be something called adenine, something called thymine, something called guanine, or something called cytosine. Your book shows you a picture of the structures. You don't have to memorize the structures, but you might want to look at the structures because on the structures you will see an area where there's an oxygen and an area where there's a hydrogen, and those are areas where you can form hydrogen bonds because that's exactly the way the hydrogen bonds form on water. And so the hydrogen bonds are going to form the same way. Now, we often abbreviate these bases with their first letter, thankfully. So there's A, T, C, and G. And we have these base pairing rules that say that these bases pair up or form hydrogen bonds in a very consistent way. A will always form hydrogen bonds with T's, and C's will always form hydrogen bonds with G's. And this structure and these base pairings basically are what lend itself to DNA being able to replicate itself so easily. So if you only had one template strand, if you only had one of these strands, you could figure out the bases in the other strand by using those base pairing rules. The order of the bases, the order of the DNA sequence, whether it goes T, C, C, T, or whether it goes T, C, G, T, or whether it goes A, C, G, T, that is going to vary among different species, different individuals. Each species has very characteristic DNA sequences. So if we're talking about canine hemoglobin versus human hemoglobin, those will have very characteristic DNA sequences. And also, if you have a different gene for a different trait, that may have a slightly different DNA sequence. So the gene for blue eyes versus the gene for green eyes are going to be very similar because they're going to code for the similar protein of melanin in the eye, but they might differ slightly in the DNA sequence. So they show you in this little red box what they mean by a base pair. And these would be held together with hydrogen bonds. And actually, there usually is 2 hydrogen bonds between A's and T's. And then there's 3 hydrogen bonds between C's and G's based on the structure of those nucleotides. But the hydrogen bonds will hold these 2 strands in place, and then they will form a helix around each other, or they will wrap around each other. Now, a cell has to replicate its DNA before it divides. Your book before this goes into a really detailed discussion of chromosomes and what chromosomes are. I'm not taking the time to go through that discussion about chromosomes because that's pretty straightforward. That's something that you can read about and read the PowerPoint and look at the animations, and you can probably pick that up yourself pretty well. So I'm not taking the time to go through that because that's pretty straightforward. But your book tells you humans have in their body cells 23 pairs of chromosomes, or 46 chromosomes. And there's a table that shows how many are in dogs and how many are in a bunch of other species. Well, all of your cells need to have those 46 chromosomes. And when a cell goes to divide, it'll still need 46 chromosomes. If it doesn't duplicate those chromosomes, then the first time the cell divides, there will be 23 chromosomes in each of the daughter cells. And the next time the cell divides, there will be 12 and a half chromosomes in each of the daughter cells. And the next time it divides, there will be 6.25 chromosomes in each of the daughter cells. And that's not the way that you want cells to divide. We need to have cells divide a lot. As our hair grows and our nails grow, cells are dividing. As you cut your skin and the cells grow to repair your skin, then that's because cells are dividing. So cells divide quite a bit. And so a cell needs to replicate its DNA or go through DNA replication before it divides. And luckily, this is a pretty easy process because of our base pairing rules. So each strand of the DNA can serve as a template. And we can synthesize a new strand of DNA, and all we have to do is follow the base pairing rules. And it'll result in two double-stranded DNA molecules identical to that original molecule that we had, so that when the cell divides, one DNA piece can go to one cell, and an exact identical DNA piece can go to the other cell. So we can do an example of this. T, C, A, C, A, T, G. So I'm just randomly making up letters here. And here I'm going to give you the base pairing rules. So when this DNA molecule, before it divides, it has to unwind, the helix has to unwind, and the helix has to separate so that there will be some space for the enzymes to work. We're going to talk about enzymes next. But the other strand of this so these two strands would be hydrogen bonded together. There would be hydrogen bonds here. And they would this would be twisted into a helix. And when it's ready to replicate, then it's going to unwind and it's going to come apart, and those hydrogen bonds are going to break. And enzymes can come in and fill in based on our base pairing rules, based on the hydrogen bonds that can form. And so based on our base pairing rules, the enzyme can fill in nucleotides that form hydrogen bonds or that match the base pairing rules to fill in the other strand. So, yeah. There we go. So an enzyme comes along and fills this in, and so what we end up with at the end of this is two DNA molecules that are exactly the same. TA, TA, CG, CG, AT, AT, CG, CG. So this DNA molecule can go to one cell. This DNA molecule can go to the other cell. And the two daughter cells have the exact same DNA that was in the parent cell. And that process is called DNA replication. And it uses the base pairing rules. It uses some enzymes. So in order for DNA to replicate, the double helix has to unwind using an enzyme called helicase. Then the nucleotides have to be filled in, and that happens with DNA polymerase. And then any gaps are sealed by ligating the pieces together. That happens by an enzyme called DNA ligase. And so it forms a continuous strand. And so those are some of the enzymes that are used to put together. Now, if my little explanation didn't really help, this is a figure from your book that shows the same thing. And so they start out with 4 nucleotides. This is the original DNA strand. And then they actually rewrite it to show you the separation between the 2 strands. There has to be space for the enzyme to come through and the nucleotides to fill in. And so each of these strands is a template. Each of these strands serves as the instructions. And we fill in with additional nucleotides. Whichever nucleotides can form hydrogen bonds. And what we end up with is 2 molecules that are exactly the same as our original molecule that we started with. GC, GC, TA, TA, AG, AG. So they're exactly the same as that original molecule that we started with. Now, because we end up with, in each of these daughter molecules, 1 strand that was from the original molecule and 1 newly synthesized strand, this is sometimes called a semi-conservative model of replication, of DNA replication. It's sometimes said that DNA replication is semi-conserved. And what that means is we keep 1 original piece, or 1 piece from the original molecule, and then 1 piece is newly synthesized. And it's that way in all of the daughter molecules. So if you ever see the term semi-conservative replication, that's what it's referring to. Now there's a couple of animations that are in the PowerPoint. If you're in slideshow mode in PowerPoint, you can click play on those, and those will show you several of those. And then here's a molecule, or here's a figure from your book showing the same thing, showing the double helix, and then it has to unwind and it has to separate so that there's room for the nucleotides and the enzymes to fill in. And then again, they show you this semi-conservative replication because part of that strand is in each of those daughter strands at the ends. Now, organisms are made up of a lot of macromolecules, and many of them are proteins. So Chapter 7 gets quite a bit into how do we make proteins? How do we have the information needed to build all of the proteins needed to make a new organism? And so I give you a little bit of an overview here. A gene is part of a DNA molecule, and it specifies an RNA or a protein product. And the information that's in the DNA is the order of the nucleotides. And that will become a little bit more, that'll make a little bit more, it'll become a little bit more apparent when we get through our discussion of transcription and translation. So once we finish talking about transcription and translation, come back to that statement and see if that makes a little more sense. Come back to that idea that the information in a DNA molecule is in the order of the nucleotides. So again, we've talked about replication. So that very first diagram that I kind of drew out for you, the piece where DNA makes a DNA copy is replication. And now we're going to get into the steps of transcription and translation, which is a way that we make a RNA copy of DNA and then a protein out of that RNA. And that process is called gene expression. The process by which the information in the DNA becomes a protein product is called gene expression. Transcription uses DNA to make an RNA, and then translation uses that RNA to make a string of amino acids or a protein. So if we think a little bit about some of the things that we've been talking about related to cells and cell organelles, we can draw a big picture of a cell. And one of the organelles inside that cell is the nucleus. So this black circle is going to represent the nucleus. And the DNA is in the nucleus. DNA is a double helix inside the nucleus. Now, DNA lives in the nucleus. DNA never gets out of the nucleus. If your DNA ever gets out of your nucleus, the cell tries to kill itself. That's how bad it is if DNA gets outside the nucleus. DNA stays inside the nucleus, except for some steps where the cell is dividing and that nucleus has to divide. Other than that, there's that double membrane nuclear envelope around the nucleus, and that contains the DNA. But the other thing that we learned when we learned about organelles is we learned that there is a membrane-bound organelle called the endoplasmic reticulum. And there's rough ER that has ribosomes on it. We also learned that there's some free ribosomes that float in the cytoplasm. And when you learned the function of ribosomes, you probably learned that ribosomes function in protein synthesis. So if the DNA contains the instructions for making the protein, but the protein is made on the rough ER or the ribosome in the cytoplasm, and the DNA never gets out of the nucleus, but proteins are made in the cytoplasm. Well, the way we can reconcile that is that we have an intermediary molecule. We have a single-stranded nucleic acid molecule called RNA. And RNA can leave the nucleus. RNA can fit through the pores in the nuclear envelope and can go to the ribosomes, either free or on the ER, and can make protein. So while replication is just a single-step process, protein synthesis has to be a two-step process. It has to have transcription and translation because of where the organelles are located in the cell. So transcription is making an RNA copy of a DNA molecule, and translation is making a protein from that RNA molecule. So the first step that we're going to look at is transcription. So the nucleotide base sequence in a gene has the instructions for building an RNA or a protein product. And the DNA is in the nucleus, but proteins get made on the ribosome. And so what happens is the cell transcribes that information into an mRNA, and the mRNA can go to the ribosome. An analogy that I use a lot is if you're building a house and you're at the construction site, but you need the blueprints, which are at the office. And so you send somebody to make a copy of the blueprints and bring it to the construction site. And so making a copy of the blueprints is transcription. You transcribe, which just means to copy. You transcribe the information that's on the blueprints into a copy to take to the construction site. Here we transcribe the information that's in DNA and then take it to the ribosome, the construction site. Now, we pretty much follow the same base pairing rules as we did for replication, with one exception. There is no T in RNA. Instead, it's replaced by a uracil, or a U. The difference between DNA nucleotides and RNA nucleotides is the sugar that's attached to them. The DNA nucleotides have a sugar called deoxyribose, which is what the D in DNA means. RNA nucleotides have a sugar called ribose, which is the R in RNA. And there is not a ribose nucleotide that has a thymine base. It just doesn't occur in nature. There is a ribose nucleotide that has a U, or a uracil, attached to it. And your book shows you the relative structures. They're very similar in structure. So our base pairing rules that we used for DNA work as far as the C and the G and the T to the A, but it doesn't work for A to T. Instead, T is replaced with U. So if these are our DNA nucleotides, when we're doing replication, when we're making a DNA copy, C pairs with G, G pairs with C, A pairs with T, and T pairs with A. Those are our base pairing rules that we had already talked about, T and A and T, C and G, G and C. But if this is our template and we're making an RNA strand, like in transcription, here C still pairs with G, G still pairs with C, but now A will pair with U because there is no T in RNA. There is A in RNA. So the only one that's going to be different is there's no T in RNA. It's replaced with U. So anywhere that there is anywhere there's a T in the DNA, there will be an A in the RNA. Any place there's a C, there will be a G. Any place there's a G, there will be a C. So all of those are going to be the same as our DNA rules. The only difference is going to be where there's an A in the DNA, there will now be a U in the RNA. Uracil replaces thymine. So transcription starts with the enzyme binding to the DNA. The enzyme is called RNA polymerase. Previously, we had an enzyme that was called DNA polymerase that made a DNA that occurred in replication. Here we have an enzyme called RNA polymerase that's involved in transcription. It makes the RNA. The polymerase moves along the DNA, it unwinds the DNA, and it puts the RNA nucleotides in their place using the base pairing rules in the order determined by the base sequence of the gene, using the base pairing rules. Then the next step is translation. Now that the mRNA is formed, and it has the same information as the original gene, it can leave the nucleus, goes to the ribosome, and basically we translate the message into a protein. We do that by using something called the genetic code. This is figure 7.5 in your book. This is the genetic code. So I want to talk a little bit about how to use this figure. The genetic code is the same for almost all organisms on our planet. So bacteria use this genetic code to make their proteins. We use this genetic code to make our proteins. And if you notice, what it shows you are 3-letter triads called codons. The information in an mRNA consists of 3 nucleotides. 3 nucleotides is a codon, and that codes for 1 amino acid. So it's a triplet or a set of 3 bases. Those 3 bases code for 1 amino acid. Now, the way scientists figured out this chart is they broke open a bunch of cells, and they figured out this process of protein-making machinery. They figured out wherever these 3 bases were in an mRNA, then this amino acid occurred in the protein. Wherever this codon was, this amino acid occurred. Wherever this codon was, this amino acid occurred. And they did that for lots of different species and lots of different cells, and they figured out that it was pretty similar. It was the same. So they came up with this genetic code. This is a codon chart. So what you're looking up here, what these 3 letters are a codon in the RNA molecule. And the way you find your codon, you can just kind of scan and read through all 64 of the codons until you find the one that you're looking for. Or you can match up the first base of your codon as one of these letters. Then the second base of your codon is one of these letters. And then you can find the third base, and that will help you locate your codon on the chart. So, for example, if the codon that we're looking for is GAU, the first base is G, the second base is A, and the third base is U, so we triangulate all those 3 to here. Or you can just scan it until you see GAU. The amino acid that corresponds to GAU, its 3-letter abbreviation is ASP. That's an asparagine. You do not have to memorize this chart. If you ever need this chart, it will be very easily accessible to you. If you need it on a test, you can use it out of your book. If you're a scientist one day and you need to know it, it's very easy to look up on the internet, and most people have one stuck to the wall of the lab. So this is not something that you're going to need to memorize. You are going to need to know how to use it. So if I ask you a question like, what amino acid corresponds to the codon UGU, then you could come find UGU, and you can tell me the corresponding amino acid is a cysteine. So those are the types of ways that we'll use the chart, and I'll show you a little bit more about that in just a minute. And you're going to need to use this chart when you do your assignment this week. So we're going to go through it again in a couple of times as practice as we're using it as part of this discussion. But you can see from this chart that one codon codes for 1 amino acid. One 3-letter sequence codes for 1 amino acid. And so this is a figure from your book that shows you In the blue is DNA, so this is a region from the gene in the DNA. And then the brown they show you is the mRNA. And that's, I think, the same sequence that they showed you in the previous figure that we were looking at. So they use the same color combinations. Here, they're actually showing you these three amino acids, these three nucleotides code for this amino acid. These three nucleotides code for this amino acid. These three nucleotides code for this amino acid. So they're showing you with these green arrows. And you can actually look these up. In fact, that might be good practice to look those up in the codon chart and practice looking up codons in the codon chart and matching them to the amino acid sequence. Now, there's a couple of different RNA molecules that we're talking about here. I gave you a summary slide a little bit earlier. There's the mRNA, which is the messenger RNA. That's the copy of the DNA. That's what we're translating here. That's what we're turning into an amino acid sequence. There's also rRNA and tRNA that help with this translation. rRNA is ribosomal RNA, and tRNA is transfer RNA. The ribosomal RNA is a part of the ribosome, so that's part of the structure that's going to be doing this. And then the tRNA matches up the codon with the amino acid. So I want to talk a little bit about the structure of tRNA for a couple minutes, because that sometimes gets a little bit confusing. Transfer RNA is a type of RNA, and there are two parts that are important. It has an area called an anticodon, and it can base pair with the codon. It can match up to the codon. And then the tRNA has a site that binds to the amino acid that's specified in that chart, that's specified for that codon. This is a figure from your book. This is actually what it would look like as a molecular model. So this is all of the nucleotides that make up the tRNA. There's only two parts of it that are important, though. There's only three bases in this whole thing that's important. And those are the three that are the anticodon. And then the other part of the molecule that's important is the amino acid. So one end has the anticodon, and one end has the amino acid. So we just kind of show the rest kind of as gobbledygook. We just kind of show the rest of the molecule, and then we show you the specific amino acid that's attached, and then we can show you the specific anticodon that would be attached. So what happens when you're doing the translation? There's a whole series of pictures that are on pages 122 and 123. For translation, all of these pieces come together. The mRNA that was made in transcription, the ribosome, and these transfer RNAs. You get two transfer RNAs right next to each other, and they can form a peptide bond. Then this amino acid, this tRNA is now empty. This tRNA goes away, and the next tRNA comes in. It forms hydrogen bonds. It base pairs with the codon, and then once it's base paired with the codon, a peptide bond can form. And now the empty one can go away, And this whole thing is shifting over. This whole ribosome is running down this mRNA, almost like threading fabric through a sewing machine. You're threading this mRNA through this ribosome like you thread fabric through a sewing machine. And just like the makes a stitch in the fabric of a sewing machine, here we're making peptide bonds. We're stitching these amino acids together. So you can form an amino acid. The whole thing moves over. One goes away. One comes in. You make a peptide bond. The whole thing moves over. One goes away. One comes in. You make a peptide bond. And that will happen over and over down the whole length of the mRNA molecule. And that's how you string together these amino acids to make your protein. And so that's the process of translation, stringing together these amino acids to make the protein. What amino acids go where? Well, that depends on the nucleotides of the codon. So I'm not done, so don't think that I'm probably going to run over the 2 o'clock hour a little bit. So it might be a little bit after 2 when we're finished. But I want to make sure that I go back and explain some things that are going to relate to the assignment. So the first thing that we talked about was replication. When a cell divides, it needs to copy its DNA. That's done in Chapter 6. That uses an enzyme called DNA polymerase that uses some of those other enzymes that were on that slide. We unwind it. We fill in with bases. But when a cell is doing some of the cell functions like transport and metabolism, it needs specific proteins, like it might need an enzyme to catalyze a reaction. It makes them by transcription and translation. We're relying on these base pairing rules in a couple of different ways. We rely on the DNA-RNA base pairing rules in transcription. We use DNA for a template, and we fill in RNA nucleotides. But also, the anticodon codon base pairs help to bring in the correct anticodon that brings in the amino acid that's next to be added. Yes, so the answer to Danielle's question is yes. The peptide bonds are made on the ribosomes. And I'm trying to see if I can find a good picture of that. So the ribosome, they show you here, it kind of has a tunnel through the middle of it. It kind of has a hole, and that's where we're going to feed some of the mRNA. And so the ribosome is going to help to function to catalyze that peptide bond. And so all of these structures are attached to the ribosome as this process is going on. So the mRNA is being fed through the subunits of the ribosome. And then the tRNAs actually base pair. They form hydrogen bonds, and they attach to the ribosome. And then that peptide bond forms. And then after that peptide bond forms, so now both of these amino acids are attached to this tRNA. This one's empty. It can go away. And the next one comes in. And when these two amino acids are close enough, a peptide bond forms here. And so now all three of these are attached to this transfer RNA, and this empty one goes away. It'll actually go pick up another amino acid so that it can participate in the process later on. and another one comes in, and when these amino acids are close enough, then the peptide bond forms. So there is this intermediary molecule that we have, this tRNA, that helps us go from the codon to the amino acid. But when we look at our codon chart, this chart is a codon chart. This chart does not show you anticodons. This chart shows you codons. So it's showing you the codon that's in the mRNA and what amino acid will be added. That just helps you not have to make it yet another step. You don't have to translate the DNA into a codon and then translate the codon into an anticodon and then look it up in the chart. You only need to know the base pairing rule and then how to use the chart. So we can use an example. Now, I normally show this slide stepwise, but I'm not able to do that in Wimba. So right now, we're only going to look at these top two lines. So here's the DNA template. Here's the DNA nucleotides that would be in that DNA. And so in the nucleus, we can make an RNA transcript just by using our base pairing rules. So C can become G, A pairs with U, T pairs with A, because there is A in RNA. There's just no T in RNA. C and G are the same, G, G, G. Again, T is replaced with U, so A would base pair with a U. Oops. And then you can take these codons and look in the codon chart and find out what amino acids will be added to the protein. So if you look on the codon chart, if you look up GUU, a valine will be added. If you look up AGC, a serine will be added. If you look up CCC, a proline will be added. So that's essentially how you're going to do your assignment. And what I have here shown in light blue is to remind you that this codon becomes this amino acid because there is an intermediary molecule. There's a tRNA that has an anticodon. And so this tRNA molecule at one end has this anticodon, and at the other end has this amino acid. All of these have this, at one end have this amino acid, and at the other end have an anticodon. And so they form base pairs with the codon, and then a peptide bond forms between the amino acids. But I include it because the other thing I want you to notice is if you look at the sequence in these anticodons, and you look at the sequence of the original gene, the original DNA template, they look very, very similar. So in this slide, when we were originally saying the information is in the order of the nucleotides, the information in the gene, in the DNA molecule, is in the order of the nucleotides. So now we can kind of come back and think a little bit about what we mean by that. The specific nucleotides that are there determine the specific nucleotide, the specific amino acid that's going to be in the protein. So a CAC in the DNA might give you something totally different than a GAC, or a CAA, or a CGC. There's a number of different ways that you can change this codon by just one letter and have lots of different amino acids be in that place. So it's not just that there's 3 of them there. So the information is not in the number of nucleotides. The information is in the order of those bases. CAC is going to be different than GAC. Is going to be different than CGC or CUC or CAU. You can think about all the different ways that you can vary 3 nucleotide sequences, and those are all the different options for amino acids that you have there. So it's not just that there's 3 of them, it's the specific 3 that are there. The information, or what amino acid goes into the protein, is specified by the order of the nucleotides, the bases that are in that codon. So that kind of makes that statement just a little bit more clarified, helps that statement make a little bit more sense. So when we take this gene, when we have information in this gene, when we have DNA sequence in this gene, and we're trying to get to a protein, and the last step is the tRNA coming in to make the protein, we have kept the information the same. We have kept the information from making a DNA copy to an RNA copy to the tRNA anticodon. The information has been returned, has been preserved. Sorry, it should be anticodon. We've kept the same information. This CAA is CAA. This GGG is GGG. AGT is the same, it's just AGU. TGC is just UGC. So we've just replaced T's with U's, but other than that, the information is the same. And so we've kept that information and gone from the nucleus to the ribosome, and we've used that information to determine what amino acids go in this sequence. Okay, so what questions do you guys have at this point? I want to go through just a little bit with mutations, but is there something that you want me to go back over before I do that with transcription and translation? Well, what we're talking about with mutations builds on what I was just talking about just varying codons by one letter can vary the amino acid that's there. If you just change one letter of a codon, you could change the amino acid that's in the protein. Mutations are going to be permanent changes. So you can have a mistake in the process of DNA replication, but those mistakes are usually corrected. That's not considered to be a mutation. A mutation is a permanent change where there's no way of knowing what that original letter was supposed to be because that cell has divided and divided and divided. A mutation that changes a protein may have harmful effects. So mutations that affect the proteins in hemoglobin might affect the body's ability to carry oxygen. Underneath the genetic code figure in your book, it gives you a list of amino acids, and it gives you their one-letter abbreviation. Some of those amino acids can be acidic, and some of those can be basic. Some are neutral. Some are charged positively. Some are charged negatively. Those amino acids affect how the protein folds. If you change that amino acid, you might change the way the protein folds. And if you change the way the protein folds, you might change its function. Folding is very important to how enzymes work. So some of this is building on some of the things that we talked about in Chapter 2 and Chapter 3 with the macromolecules and the protein folding, and then some of the things with the enzymes or those kinds of things. By changing the amino acids, we can change the folding. By changing the folding, we can change the function. So if you change a nucleotide, you could change an amino acid, and you could change that whole protein's function by just changing one nucleotide. So that's why mutations are such a big deal. So your book gives you several examples of mutations. So this is the DNA that they've been using, shows you the RNA, shows you the amino acid. So you can use this to practice. If you cover up this mRNA part and you just use this DNA part, see if you can use the base pairing rules and see if you come up with the same thing. And then use the codon chart and see if you use these codons, do you come up with the same amino acid? And check your work. But this is the normal amino acid sequence. What happens if we have a small deletion in the DNA? Here we've deleted a single base pair. And since it's every three nucleotides that code for an amino acid, we've altered the whole rest of this amino acid sequence just by deleting a single base pair. The same thing can happen if we just alter a single base pair, if we just have a substitution where one base is replaced with another. That just affects this one amino acid, but if it's a charged amino acid in the center of the protein, it might totally alter the way that protein folds. It might push some of the other amino acids toward the outside of that structure or that folding. So even after looking at mutations, we can go back to the sentence. The information is in the order of the nucleotides. If you change the order of the nucleotides, if you change the letters that are there, you change the information. You change what amino acids are going to be in the protein. So your book gives you this sentence. The information is in the order of the nucleotides. But it sometimes doesn't make sense until after you've gone through some of the other information and understand it, that you really understand sentences like the information is in the order of the nucleotides. But then once you understand a little bit more about transcription and translation in the process, the way that you have to use those three nucleotides in the codon to code for one amino acid, then that makes sense. Statements like that make sense. Okay, so now I'm on the last summary slide for real. Other questions that you guys have about transcription, translation, or anything in 6 and 7? Well, I went over a little bit, but I knew I would because I wanted to make sure and explain some of those things that wouldn't be as evident from doing the reading. And I'm hoping that by giving you that example, that might help with what you're working on for the assignment this week. So, as usual, if there's any questions or anything, shoot me an email or post it up to the discussion board.